that make that happen in space and time. At UC Santa Cruz, I totally focused on C. elegans, and then I went to Stanford University for a couple of years to work on a different system, which is basically dissociated neurons from the rat brain. So essentially we take a rat brain, dissect it, break it down into its component cells, and culture them in an in vitro system. And then using that, those cells will naturally form synapses or connections amongst themselves. And I did a lot of microscopy and imaging study of those systems. So then when I came to the U, I went back to worms primarily. And so what I'm going to do today is, I thought the best way this talk could help you guys in your curriculum development would be essentially to explain why we use worms. So the reason I started doing stuff like this was about almost 4 years ago, 3 years ago, there was a presidential election, and it was between McCain and Obama. And one of the contentious issues that came up that Senator McCain brought up, not in a very well-publicized debate, that I thought was embodying a big problem that I had to, at least on an individual level, try to strike out and deal with, was Senator McCain was reading basically NIH and NSF grants. And he was making some very derogatory statements, one of which was, why are we funding research on grizzly bears and flies? So that's a statement of profound ignorance. And unfortunately, I have to say that. I mean, I have nothing against Senator McCain, I actually kind of like him, but that was a galvanizing moment for me, where I realized that, one, I like grizzly bears, so there's a misunderstanding about evolutionary and environmental and biology and ecology that's taking place there. There's also a more fundamental problem, because the fly disconnect is more, it's a worse problem, because that means that certain people in the government don't understand the connection between the fruit fly Drosophila and some of the biggest medical discoveries that have ever been made. So for me, that's a big problem. Obviously, from the fly, the implication would be worms are less important, because we as human beings tend to be anthropocentric, tend to be fixated with ourselves. So one of my mentors often said worms are more evolved than human beings and more evolved than flies, because their generation times are shorter. And I stand by that. So from an evolutionary perspective, they're actually more advanced than we are. And they do what they do very, very well. But we still have to deal with that issue. And one of the best ways I thought to get that out there was to start to interact with students at all levels of education. So I've done elementary stuff all the way through high school. And particularly try to reach out at the high school level where more complex concepts can be conveyed. To get people to understand this idea that there isn't really a disconnect between studying certain concepts in something like C. elegans versus studying it in a human being. There's more similarity than you would imagine, and that's what I'm going to try to convey today. And I think that's a very important concept for students when they want to understand how neuroscience works. The first reaction is to think, how does my brain work? I think that's an important question. That's like the fundamental question of the whole center of neuroscience. But I think a concept that rarely gets conveyed is that the complex system isn't the best one to understand. And the example I use is always the computer. If I were to give you a gigantic computer, let's say a multi-server computer with 50 terabytes of capacity, good luck trying to figure out how that works. Have fun, especially if you don't know, because assume we don't know, because that's how I operate on a daily basis, we don't know. It's a nightmare. C. elegans, by comparison, would be like taking something that runs off the same components, but much simpler. So the same machinery, the hardware is the same. It's just that you have way fewer connections to deal with. And so understanding it becomes a much simpler task. Okay, so what am I now? So now I'm an assistant professor at the University of Minnesota. I'm in the pharmacology department, which seems like a disconnect, because I'm technically a molecular neuroscientist. The way I explain that to people is that pharmacology is a study of drugs, essentially, and how they work. Any person who studies cellular signaling, which is how information goes from the outside of a cell to the inside of a cell, is essentially working on drug targets. And so that's what my lab focuses on. In worms, what we really use a lot, is, we focus on certain particular pathways that I'm not going to talk about today that are particularly amenable, that are related to regeneration, nervous regeneration, and that are also related to synaptic development and neurodegeneration. So that's kind of my focus. But I think what will help is to start at the beginning with a very big idea, So essentially, you're all familiar with the human brain. And the way I think about the human brain is in two ways. Human brain in health, human brain in dysfunction. Not disease, because I think that's maybe too strong a word. So over here, you can think about all the things in health that come with your brain. Your ability to learn and remember things, your ability to perceive and respond to your environment. And of course, that is essentially what makes up who you are. It's this interplay between these things that creates your personality and everything that comes with being a human being. Okay, so when we think from a medical perspective about what happens in terms of, actually, can I get rid of this little one? Let's see here. Okay, so when you think about the medical perspective and what one would study, what are the relevant questions medically? And I think a high school student is interested in these kind of things. When you think about what makes up a human being and your mind, the thing that comes right along with that is what are the problems that go wrong and how does that affect people? So the most classic one that most people have heard of is neurodegeneration. Most everybody knows somebody who has Alzheimer's disease or Parkinson's. Those are widespread diseases, late onset, so always in predominantly older people. The younger you are when you get it, the worse it's going to be. Pretty simple. The fundamental part about all these diseases, Huntington's, ALS, which is Lou Gehrig's disease, they all have a central problem, which is your neurons cease to function. Whether they stop forming connections properly, whether they stop sending signals properly through your brain or through your muscles, your neurons to your muscles. In either case, you run into a catastrophic disease state. So by understanding how a synapse forms in time and space, and the triggers that regulate that process, we hope to work on trying to help something like this, okay? With a more broad-based therapy, not a disease-specific therapy. So then there's the traumatic injuries to the central nervous system, hitting your head on the pool bottom and breaking your neck. We've noticed a lot of increase in traumatic brain injury, so war-related injuries, often central nervous system trauma-based a lot of the time. Those things are basically necrotic damage. In that case, you're not seeing the cell sort of fall apart from the inside out, like in a neurodegeneration case. You've got a traumatic event that triggers cell death, and that causes damage, and that leads to a loss of function. So one of the fundamental questions right now in neuroscience is, if somebody walks in who's paralyzed due to an injury, we can't regrow the neurons. It's not possible. The closer you get to the central nervous system, the harder it is. So peripheral neurons can regenerate a little bit. When you get into the spinal cord and the brain, they really don't very much. And there's lots of reasons around that, and we can talk about that. But essentially, that's a huge goal for medicine, basically, is can you trigger regeneration? Okay, so that's the other sort of therapy associated with studying neuroscience. And then the other one is, I don't know if you guys can see this that well, but it's developmental defects, genetic developmental defects. That's a totally different class of disease. So that's people who are born with a problem, Down syndrome, autism, and it can manifest and become worse over time, okay? And in a lot of these cases, you have essentially a problem with synaptic wiring, neuronal wiring, okay? So one thing I should say, I want to make this as informal as possible, so if you have any questions, just jump in. It's the first time I've given a talk to an audience like this, so if certain concepts aren't making sense in any way, just ask as many questions as you want. I don't really have a time frame, so we can kind of just bounce it around and see if we can make sure things are clear, and then that'll help me improve for the future. Okay, so the place I want to start is, I think a lot of people don't know what a synapse even necessarily is, so we're going to start with the basic. So, in your brain, there's billions of neurons, basically, and they're all connected to one another in different cell types, okay? The basic idea behind a synapse is very simple. You have two neurons, so imagine we zoomed way in on this brain, into a very tiny section of the brain. And I have cell here and cell here, okay? Two cells. They could be very far apart spatially, so don't assume because they look close here, they're close. They could be very far in distance. Essentially, a neuron has certain features, okay? It's got a cell body, and this little red thing is the cell nucleus. It's got what are called dendrites. So these are basically polarized pieces of the cell that extend off, but they're not super long, and it has a lot of them. The neuron has many dendrites, and that's where it receives its input, okay? Then what it has is a single long axon. Sometimes it's myelinated, like this red stuff. You may have heard of myelin. That would be a peripheral neuron that's myelinated generally. And so what you see is where these two neurons, axon connects to dendrite, that is a synapse basically. A pre and postsynaptic component come together in space and time, and they basically send signals across that junction. So the junction, what does it look like? Oops. So if you zoom in even closer, well, this is just like a cartoon here, a little schematic. This is real electron microscopy over here. So I'm going to start over here, and then I'll kind of relate it back to here. So essentially now, if we're over here, this axonal component right here that grows up, you see how it splits? That doesn't always happen, but anyways, so it grows up and makes this connection. That's this part of the neuron right here. These branches are postsynaptic, okay? And that's shown over here. Now remember, there could be an axon coming up to this cell as well, and there would be if it has a bunch of dendrites. So don't think of it as a one-to-one contact, think of it as a network. Okay, so what happens is that we have a collection of essentially vesicles. Little pieces, I don't even know how to describe what a vesicle is, but think of it as a container full of chemicals, okay? And those vesicles can fuse with the membrane that makes up the outside of the cell, and they spit out these little chemicals, neurotransmitters, okay? And that's how the signal gets across this cleft to the postsynaptic part of the synapse, okay? And this is just the idea here with the little neurotransmitters coming out. So a lot of people, you hear about these in the news and the media, serotonin, dopamine. Dopamine's the one that I think most people are familiar with because that's really associated with drug addiction and reward behavior. So basically, different types of neurons use different neurotransmitters to convey signals throughout your body. Sometimes they stimulate, sometimes they inhibit. So even though the transmitter's getting sent across, it could tell the other cell to be quiet. And it's a balance between all these signals coming in and leaving this neuron that creates information in your brain. Okay, oh, so this guy on the side. So this is just actually an electron micrograph of a synapse. And so it doesn't really look like this. It looks a lot more like this. So all these gray circles, these are the green synaptic vesicles that contain the neurotransmitters, okay? This is the synapse right here. So what you have is a pile of vesicles right near the site, and they're going to be fusing very quickly and releasing neurotransmitters. So this gray line is the presynaptic site, and that's the postsynaptic site. The other dark line on the other side, this little bit of clear space in the middle, that's the cleft, that's the gap between the two cells that the chemicals fuse across, or diffuse across. Okay, so that's essentially, there are receptors over here on the postsynaptic site, specialized proteins that can sense the presence of neurotransmitters. And that's how the signal moves across. Because this is the big problem, right? If you have this collection of cells, you've got to get information from one cell to the next. And you've got to do it using this intermediate, this neurotransmitter. And so that's essentially how it happens.